All right, this chapter is called The Chromosomal Basis of Inheritance, and this one is going to talk a little bit more about sex-linked genes and some chromosomal problems. A little history. Mendel published his papers in the 1860s, and like I told you before, he was kind of turned away by the scientific community, so he never really got justification in his contributions to science. Around 15 years later, mitosis was described, the process of cell division, and then about another 15 years later, in 1890 or so, meiosis was described, the concept of producing haploid gametes, ova and sperm. Now, the scientific community was having a problem understanding some of the activities of cells, and so scientists were gleaning through past publications, and around 1900, several of them came across some of Mendel's works. His studies in the pea plants and others gave information that the current day scientists were trying to find answers for. His publications in genetics was converged with cytology, the study of the cell, and in 1902 it converged together to become genetics, which is also diversified. Now we have chromosomal genetics. Chromosomes and genes are both paired and diploid cells. Well, this was some of the information that came from Mendel's work. Even though he didn't know about chromosomes and genes, he did know about traits. And his descriptions of the traits being passed helped the current day scientists understand a little bit more about how the cell functioned. Homologous chromosomes and alleles pair segregate in meiosis. Well, this was something that Mendel had also implied, although he didn't know about chromosomes and alleles, but he did know about traits. Both parents contributed traits to their offsprings, and so that helped the scientists to understand that there was something in the chromosomal material that was passing traits to the offspring. Fertilization restores the paired condition for chromosomes and genes. Well, the diploid cells, the body cells, somatic cells of the body all have a diploid number, the 2n number, which is 46 chromosomes. Eggs and sperm, remember they are formed by meiosis, are haploid, and they have the n number, which is 23 chromosomes. When a sperm with 23 chromosomes in its nucleus fertilizes an egg with 23 chromosomes in its nucleus, the two nuclei combine and form a diploid nucleus. That's why the egg is no longer called an egg, it's called a zygote, because it's diploid. Genes and chromosomes. Well, this says genes located on chromosomes early 1900s. Thomas Morgan was a scientist that was trying to find the first mutation, and he wanted to be the first one who could label the first mutation with some type of a symbol. And so he started the genetic symbols based on the first mutant. Well, he wanted to find one in fruit flies, and he was trying to find some mutation in fruit flies, and he wasn't finding any. And so when he was about to stop, one of his graduate students brought in some white-eyed flies. Now, the fruit flies that he had were all red eyes. Now, this was a white-eyed fly. He thought, I finally found a mutation. And so he designated white eyes with a W. And that would be X with a superscript W. And the normal eyes was going to be X with a W plus. So the superscript plus sign with a W. The example here, W plus for the wild type, that would be X, and then the superscript would be W plus. And white-eyed for the white-eyed flies would be X with a W superscript. Well, he finally found out that he had not found a mutation, but he had discovered something much more important called sex-linked genes. That the genes were located on the sex chromosome, the X chromosome. Here's a picture of the two flies. The wild type, the XW plus on the left, really brilliant red eyes, compared to the white eyes, XW, which you can see they're white. And when they're together, it's like little white pearls walking around with all the little red-eyed flies. They stand out very abruptly. White is very like bleached white. He crossed the white-eyed males. Now that's something because the white-eyed flies that were brought to him were males. There were no red-eyed, no white-eyed females brought to the professor. They were all white-eyed males. So he crossed the white-eyed males with the red-eyed females. And the F1 were all red-eyed. He crossed two F1s to get the F2, and he had 3 red-eyed flies to 1 white-eyed fly. Well, all the white-eyed flies were males. He determined that the gene for the eye color was located on the X chromosome, and I'll show you on the next slide how he did that. Females have 2 X chromosomes. Males have 1 X and 1 Y. Now on this sex-linked eye color picture here, you see that the upper left, you have the female here, and there's 2 red bars there. Those are the X chromosomes, so they're just using bars for the X. And you see that the W plus is on both of them. So she's red-eyed for sure. Well, look at the male. His X chromosome has a W, and then that little hooked chromosome there is the Y chromosome. Now these are the parents that he crossed initially, the white-eyed males that he was given with the red-eyed females. You can see that since both of these X's have W plus, instead of putting 2 of them here, you can just use 1. They're the same thing. Now, in this Punnett square, you put the female on the side, and you put the male's gametes he can produce on top. He can produce half of his gametes are going to have XW, half will be Y. And you do your Punnett square, and you see that XW plus XW, red-eyed female. XW plus Y, red-eyed male. XW plus gives you the red eyes. Well, he crossed one of these red-eyed females with the red-eyed males, and you see the gametes the female can produce through 2 different X's now. One will be XW plus, and the other is XW for white eyes. The male produces 2 types of gametes also, XW plus and Y. Now when you do the Punnett square, you see XW plus, and you see that that's a red-eyed female. XW plus XW, that's still a red-eyed female. So as long as the female has 1 XW plus, 1 red-eyed gene for the X chromosome, she's going to be red-eyed. Let's look at the males. XW plus Y, red-eyed male. He only has 1 X chromosome, and this is for red eyes. XW, Y, and there's a white-eyed male. Now when you're doing sex-linked problems, you actually have 2 things you have to pay attention to. One of them is the trait being studied, and on this one it's the color of the eyes. The other is the gender, male or female. Now in a problem like this, which you can see is worked over here on the right also, but you can't tell male and female on the right one because there's no X's and X's. Well, yes, you can. There's a Y here, but it's better if you put the X's on there. So if you said what proportion of the offspring could be white-eyed, okay, that's going to be 25%. Look over here on the left-hand side, it's 25% of these flies are white-eyed. What percentage of the males could be white-eyed? You're just talking about the males now. That's going to be 50%. What portion of the females can be red-eyed? 50%. What portion of the female flies are red-eyed? 100%. What's the chance of getting a white-eyed female? 0%. So see it, both things, the trait and the gender are considered in sex-linked problems. Linked genes. This refers to genes located on the same chromosome. That's like chromosome 1, let's say, would have genes A, B, C, D, E, F, G. And its homologue would have the same sequence, A, B, C, D, E, F, G. So those are linked in order from A, B, C, D, E, F to G. So they're linked. Now during crossing over, linked genes do not normally assort independently of each other. That means that A will not go over, A will not be crossed over between the two non-sisters. It's going to be a length of them, A, B, C, D, which is what it says here. Linked genes usually assort together. A, B, C, D, E is changed with A, B, C, D, E. Now, independent assortment of chromosomes and unlinked genes. Remember, independent assortment's random. And so crossing over is going to occur. Now, unlinked genes, these are ones that are on non-homologous chromosomes. That does not happen. Here's crossing over. And you can see that they've already formed their synapses and the chiasmata, and crossing over has already occurred here because you have exchanges between the red and the blue chromosomes. If this blue one was dad's X chromosome and this red one was mom's, or this chromosome in general, they've exchanged pieces during crossing over. And so at the end of meiosis 1, you have two cells that are haploid. One's getting this chromosome, one's getting this one. And when they complete meiosis, you end up with 4 different gametes. One that's all blue, one that's red, blue, red, one that's blue, red, blue, and one that's all red. So over here it says that crossing over occurs between non-sister chromatids, which you can see there, non-sisters, and crossing over increases genetic diversity. It mixes the DNA, so you get a greater diversity or outcome. Like your brothers and sisters may look like you, but you're not identical. Here is sex determination. Like I told you earlier, females have 2 X's, males have an X and a Y. So the Y is what makes males male. Now, the chance of having a male or a female, probability-wise, you do a Punnett square, you can find out real easy. Now, I just have 1 X here, but you could put 2 of them here, that's fine. Because females make 2 gametes, but they all have the X chromosome. Males can produce 2 different types of gametes. Half will have X and half will have Y. So you put the male up here at the top. You can put the female down here. And when you do the crosses, you get XX, XX, XY, XY. That's 2 females to 2 males. Well, you reduce to the lowest common denominator, which is 1 to 1. 1 male to 1 female. So the chance of having a male at any one point in childbirth is 50%. 50% chance for having a female. X-linked inheritance. Okay, and man involves genes located on the X chromosome. That's why it's called sex-linked or X-linked. An example of X-linked recessive characteristics are red-green color blindness and hemophilia. Even pattern baldness is a sex-linked trait. If the female is heterozygous, that means she has 1 normal and 1 affected X. For an X-linked recessive trait, she is normal, but can pass her gene to her offspring. If she passes it to a son, the son will express the recessive trait because he only has 1 X chromosome, which he receives from his mother. So if the mother's heterozygous, the son has a 50-50 chance of being normal or having the X-linked recessive problem. Here's an X-linked problem, like you would get on a test or something. In man, red-green color blindness is X-linked. Normal color vision X big N, which I would put off to the side, I would put X big N equals normal color vision, is dominant to color blindness, X little n. I'll put off to the side, X little n is colorblind. So you have your code off to the side on your paper. A man with normal color vision, so you know he only has one X, so he's got X big N, Y. That's what it shows down here. He has normal color vision, so you know what he is. Marries a woman with normal color vision. Well, she could be X big N, or X big N, X little n. So let's see what it says. Her father was colorblind. Well, her father had the X little n, so that's what he gave her. And if she has normal vision, mom gave the X big N. So here is the woman down here. It says show the genotype of the man and woman and the genotype and phenotype of their possible children. Well, here's the man, X big N, Y. Here's the female, X big N, X little n. And she still has normal color vision. Here's the problem. You're making a Punnett square. So here's the female, X big N, X little n. So she can produce two different types of gametes. Half will have X big N, half will have X little n. The male, X big N, Y, he can produce two different gametes. Half will have X big N, half will have Y. So put the male across the top, X big N and Y. Put the female down the side, X big N, X little n. Now when you do the cross, you have X big N, X big N, X big N, X little n, X big N, Y, X little n, Y. Now that's going to be your genotypic ratio. I just don't have it shown here. It'll be 1 X big N to 1 X big N, X little n to 1 X big N, Y to 1 X little n, Y. Now the phenotypic ratio, what you're going to see is 2 normal females because both of these have normal color vision. 1 normal vision male, X big N, Y, and 1 colorblind male, X little n, Y. X inactivation in females. Yeah, females are a mosaic of the X's of their parents. See, it says only 1 functional X chromosome in each of most mammalian females. Now, one of those X chromosomes has been coiled up into what's called a Barr body, and a man named Louis, it's named after a man named Louis Barr. Condensed inactivated X chromosome, and most of the genes are inactive on that X chromosome. A lady named Mary Lyon figured this process out. It's called lionization. Females, a mosaic of 2 types of cells. So one of the X's is condensed into a Barr body, and the other one is active. Now don't worry about this DNA methylation. This XIST gene produces an RNA molecule that is responsible for forming the Barr body on one of the X's. It's a non-functional RNA transcript that causes the Barr body to be formed, but it's only on one of the 2 X's. Abnormal chromosome number. First definition is non-disjunction. Non-disjunction is an unequal distribution of chromosomes between daughter cells. Now, non-disjunction is typically going to occur in anaphase because that's when, in mitosis, that's when the sisters are separated, and in meiosis, that's when the homologs are separated in anaphase 1, and the sisters are separated in anaphase 2. If something happens and they're not separated, then too many chromosomes goes to one daughter cell. So it can happen in meiosis or mitosis, and it's going to be an anaphase. Aneuploidy is extra or missing chromosomes. Trisomic means 3 bodies, so it has 3, like trisomy 21, which is Down syndrome. Monosomic means 1 body, so you're missing 1. And so there's a thing we'll look at in a minute called monosomy X. Polyploidy means many sets, so more than 2 sets. So triploid, that's 3 sets. Tetraploid, that's 4 sets. Strawberries are octoploid, that's 8 sets. Now that can happen in plants and they can still live. Animals do not live, the animal cells will not live with polyploidy. Anything more than diploid, 2 sets, and the cells die. So some problems that could occur during crossing over. There's some problems here. One's called deletion, where a portion of a chromosome is lost from the genome. These genes are not present in the gamete. The loss of genes from a genotype is serious and oftentimes lethal. So during crossing over, one of the two sisters or non-sisters lost the piece, the segment that was supposed to be crossed over. Duplication. In this situation, a portion of a chromosome breaks and attaches to its homologue. Thus, those genes are represented twice on the chromosome carrying the duplicated segment. Having an abnormal number of genes is often lethal. So you see you have a deletion here, which is oftentimes lethal, and a duplication here, which can also be a lethal problem. Inversion, where the crossing over occurs but the genes are put in backwards. Is an inversion. A section of the chromosome breaks and rejoins with the same chromosome but in a reversed order. The proper sequence of genes is often important in the coordination of metabolic pathways because the cell is very orderly. Everything happens in an order, and so if something's backwards, it will definitely affect the cell. If the inversion affects such a pathway, it can have a serious impact. Yes, it could be a lethal impact also. Translocation. That means to be found somewhere else. In a translocation, a portion of a chromosome breaks and rejoins with a non-homologous chromosome. A non-homologous chromosome. This type of abnormality can result in an increased number of genes in a gamete or can impact coordination of a metabolic path. That's going to involve too many things. Deletion, translocation. So this is oftentimes lethal also. So these are all bad crossing over problems. Now here is Down syndrome. This is caused by non-disjunction, unequal distribution of chromosomes, a condition caused by a third chromosome of the 21st pair. That's why it's also called trisomy 21, 3 bodies of chromosome number 21. This results in mental and physical handicaps characteristic of the condition. The extra chromosome usually results from non-disjunction in the formation of the egg. Now it could happen in the sperm also, but typically they'll blame it on the egg because females are born with all the eggs they're going to have. And so if a female is 20, her eggs are 20 years old, and they're only at the end of meiosis 1. So when an egg develops, it has to finish meiosis. If she's 40, that egg's been at the end of meiosis 1 for 40 years. And so things can happen to the eggs, and so they typically say mostly the problem is going to be contributed to the egg. Now, if you look down here at the bottom, you see there's the 3 chromosomes on 21, and it is a female, 2 X's. Turner syndrome, another name is monosomy X, 1 body, 1 X chromosome. Since the individual has only 1 X chromosome, the genotype is XO, the O meaning a vacant spot there. The individual is phenotypically female, but is usually sterile, not always, and has poorly developed secondary sex characteristics, kind of reduced presence. This condition does not produce mental retardation. Now you can look down here and you see she only has, on this karyotype, 1 X chromosome. Klinefelters, this is something that affects guys. XXY. Genotype results from non-disjunction, obviously. How else would he have gotten 2 X's of the X chromosome during the formation of the egg? Again, because the eggs are as old as the female. Phenotypically male, okay, because he has a Y, but sterile, so he is not fertile, and often with some secondary breast development. Affected individuals have normal intelligence. Now look down here. This guy is not just XXY. He's XXXXY. He's got 4 X's. So obvious non-disjunction. XYY and XXX. The XYY, that's going to be non-disjunction in the male for sure. Male, usually taller than normal, but are essentially normal males phenotypically. Some of your basketball players that are like 7'3" or high, taller, probably are XYY. Females, XXX. Normal, healthy, about 1 in 1,000 births can be this. They typically are a little bit taller, and you can look up any problems they may have.